The first animal phyla that we're going to talk about is Phylum Porifera. And Porifera is all of our sponges, as you might be able to tell from this comic. I'm not really going to explain the comic, so if you don't get it, it's cool. But I'll try to do that with all of our videos. So before we get started about talking about Phylum Porifera, I wanted to show you this general classification and evolutionary history of animals in general. Now you might think, oh, well, it's kind of blocked, and it is. You're going to see this again a lot. So all of the different animal phyla are going to start with the same exact phylogenetic tree. So let me go ahead and kind of trace where we are with you. So we have just animals in general. They refer to it as metazoa. Don't really worry about that. Just know this is the branch of our tree that is starting our tree. And here we have an initial divergence. We have some common ancestor that diverges into a group called Parazoa and a group called Eumatozoa. And I don't really care if you know those two terms. If you want to write them down, you can. I'm not going to test you on it. But if we go down to Parazoa, this is essentially referring to organisms with no tissues. And on this line, all by itself, is Phylum Porifera, or our sponges. Sponges are the weird one. As you can see from this phylogenetic tree, they diverged very early from the common ancestor and didn't really change much. They've been around for a freaking long time and pretty primitive. It's really easy to think of primitive as a negative thing or as a bad thing, but keep in mind we still have sponges. So even if they're not super complex, that doesn't mean they're not like good at existing because they still exist. So while I'll use the term primitive, I mean it more of they diverged earliest. They haven't derived as many traits from the common ancestor as, say, other organisms have. So let's go ahead and explore Porifera a little bit more. So, again, these are our sponges. They are not the kind of sponge you buy at the grocery store, the ones that are free colors. Those sponges are artificially made in a lab. However, if you do go to like Bed Bath & Beyond or Bath & Body Works or I don't know, like it's fancier places. You're like, there's much fancier places than Bath & Body Works. I don't know. Anyway, if you buy like, not loofahs, but they call them, they'll be like sponges and they kind of look like brownish in color. That actually might be a real sponge, like a real organism sponge. It's dead, but that is very possible a real sponge. But sponges are aquatic organisms. So once they've been out of the air for long enough, like it's dead. So if you do get one of those fancy sponges, just keep in mind you're bathing yourself with a dead animal. Yep. So just keep that in mind. That's all. So anyway, our sponges. I promised you guys that I would use a lot of those keywords in talking about the characteristics of our different animal phyla. However, because our sponges are so primitive, they actually lack a lot of things. So first of all, they lack symmetry. We would refer to these guys as asymmetrical. So they're not radial, they ain't bilateral, they're just doing their own thing. They actually don't have germ layers. There's not an endoderm or ectoderm or mesoderm because they don't have tissues. They don't have muscle or bone or blood or anything like that. Like they don't have any of that. Because they don't have germ layers, like you wouldn't be like, oh, well that means they're acoelomates. No. They're not a triploblast. They're not they're not any blast. They don't have any germ layers. Also, they don't have a digestive system. So you can't be like, oh, complete or incomplete or protostome or deuterostome because they don't have a digestive system. Now you're like, okay, how is this even an animal? So it does have digestion, but it happens on a cell-by-cell basis. So imagine, if you will, your body, you eat food and like your tongue cell eats that food individually and like gets energy, and like your toe cell eats that food and gets energy independently, and these are really horrible examples, but the point is like they don't have a digestive system that kind of breaks things down and transfers energy and transfers minerals. Instead, just each individual cell has its job of eating and getting the energy it needs. So it's still an animal, it's still heterotrophic, it's just They're simple. Again, don't think of simple as negative. They still exist, and there is a great variety of sponges. So, sorry we didn't really get to use any of those words that you just learned about, because they just lack all of that. But let's talk about what they do have instead of focusing on all the things they don't have. So they're made of a lot of different things, but we're going to talk about the two major components of sponges. So the first thing is spongin. When you think of a sponge, you're probably actually thinking of spongin. If you were to hold a sponge, even if it was a fake sponge, you feel like the squishy material. Spongin is that squishy material. Spongin is a protein. This is why that you can have a fancy sponge like you see in this picture that you're using for your hands or for your body, and it still exists. So the spongin part of a sponge is not actually a living organism. It's a protein. You could think about it like your hair. This is going to be really weird, but if you were to cut your hair and you saved your hair would always exist. Yes, it can break down over time, but it's not like an organic tissue. Like, this is all protein. I'm going to make it real gross real fast. If you're to take a section of your skin and cut it out and leave it sitting there, it would start shriveling and decomposing because it's a good organic material, lots of decomposers. So spongin is a protein like your hair. The other thing that you don't really think of, but you may have noticed, is that sponges aren't like a super soft thing. And if you've ever felt up real sponges, you'll notice they have different textures. Some of them are literally harder than others. And that's because within the spongin is a structure called spicules. Spicules look like what you see up here on the top right. They're kind of like, I don't know if you're familiar with like a ball and jacks, but they kind of look like the jacks, the pointy things. Now those spicules are made out of, it depends on the sponge, but either silica, we've talked about silica before, we're talking about diatoms, remember it's like a glass-like substance, or calcium carbonate. You may have heard of calcium carbonate before, but this is something that's found in shells, for example. So the spicules, no matter what they're made out of, is a hard material. So sponges that have a lot of spicules, when you go to squeeze them, like it's very tough feeling. There's some sponges that don't have a lot and they're softer in feeling. These spicules serve a couple different functions. So one, a structural function. If your whole body was just made out of soft mush, you would just be a puddle, like on the ground. So spicules kind of help to give shape and give body structure. Also, because they're very literally hard material, and looking at this picture, eating them probably doesn't feel too great. So spicules also serve as kind of a defensive, but not maneuver, but a defensive structure within that spongin. So this is something created by the sponge the animal, but both spongin and spicules are non-living parts of the sponge. This would be the hair that's on a human. This would be the nails that's on a human. Our hair and our nails are not living parts of us. Same thing with sponges. The spicules and the spongin itself is not a living part. Now you might be like, so like if the sponge part of a sponge isn't living, then what makes a sponge alive? I'm so glad you asked that. So let's talk about the anatomy. And when we talk about the anatomy, we're going to kind of talk about how it eats and how it moves water and the different parts of it kind of all in one. So I think I'm just going to put actually all of my bullet points up here at once. And then I'm going to kind of jump around. And well, let's actually start with the choanocytes. Now we've talked about choanocytes before when we talked about the evolutionary history of animals. That choanocytes are part of sponges. So here's a picture of a sponge. It's got a lot of spongin, it's got spicules, but in the spongin, or I should say lining the outside of the spongin, are the choanocytes. The choanocytes are the living cells. And just to show you this image again, the one in orange, that would be a choanocyte cell. So this is a cell found in the sponge, just like we have stomach cells and we have muscle cells and heart cells. Well, these are the choanocyte cells in our sponges. Now these choanocyte cells, what they do is they kind of embed themselves in the sponge, And when they embed themselves in that sponge, what they do, so this is not quite the right word, but this head-like area is embedded into the spongin. And then this flagella and this outside called a collar cell, this flagella and collar part of the cell is kind of hanging out on the outside. So the head is embedded in the spongin. That's what you're seeing here in this light tan area. And then the flagella is kind of chilling on the outside. And so what happens is this flagella is going to go back and forth. It's kind of like a rudder. And what that flagella going back and forth is going to do is literally create a water current. It's going to push water back and forth, creating this water current, which is, I don't know, I just find it fascinating. I find everything fascinating, but it's all fascinating. Anyway, creates this water current because in a sponge, there's a lot of holes in it. And we're going to talk more about that in a second. Now, as it's moving water, it's going to catch food and it's going to eat it. It's going to digest it. It's going to get the energy from it on a cell-by-cell basis. So each choanocyte cell is responsible for getting its own food. There is not going to be food delivered to it. There's not going to be energy delivered to it or glucose delivered to it that we do see, say, in humans. Each cell is responsible for getting the nutrition it needs. So let's talk a little bit more about sponges and kind of their anatomy. So if you've ever looked at a sponge, no matter what type of sponge it is, it has tons of holes in it. So we're actually going to start with all the holes that you see like on the outside of the sponge. There are small holes all over the bottom, the sides, everywhere. A single hole is called an ostium, but sponges never have just one. They have lots of them. They have hundreds of them. So you're going to hear me say ostia. That's just the plural form of it. And this is where water is getting sucked into the sponge. So all of these choanocyte cells are lining these ostia. Remember, their heads are kind of embedded in the spongin, but their flagella are out in the open. They're out in the ostia. And they're going to be beating back and forth to literally bring in water. Now, the reason it's bringing in water is because we need food. Sponges do not swim. Sponges grow like that, and they stay there. But they need food. They need nutrition. So what happens is they're creating this current, water's coming through it, and there might be some algae in it. There might be some bacteria in it. There might be some other protists. There might be things in that water. And those choanocyte cells are going to catch those things and eat those things. So this is why the water current is being created. It's essentially so those choanocytes can filter that water and take out the stuff that they can use for food. So again, they're beating in this water. So they're literally moving this water into the sponge. And you can see that with all of these little blue arrows kind of indicating water going in. And then that water now needs to leave the sponge, right? It comes in, now it needs to go out. And it goes out through an osculum. So an osculum, like you see here, is just another hole, but it's usually a much larger hole, or at least larger than the ostia. It depends on the sponge. Some sponges have really large osculums. Some of them have really small ones. It's so species dependent, but it's obvious to tell which one it is because it's the largest one that you see in a particular sponge. So again, the ostia is where water's coming in, and the osculum is usually at the top of the sponge. It's where the water is going out. And it's the choanocytes that are kind of beating back and forth that are going to move that water through the sponge. Now, before you guys move on, what I'd like you to do is actually watch the video that's linked here. It's a pretty long video, and I would recommend just jumping to the 4:21 mark in the video. One is going to show you just an animation of just how the sponge anatomy works and how these choanocytes move back and forth, which I find pretty helpful. But then it also shows a diver who is using this a neon dye around the base of a sponge, and you can get an idea of how quickly sponges can actually filter water, which I think is, it's just fascinating. It's literally like animation and then like 2 minutes worth of this neon dye, which I think is cool. So you can watch the whole video if you want to, but the most important stuff starts at 4:21, and it'll be pretty obvious when you're like, oh, they're not talking about sponges anymore. You can stop it then, or you can keep watching. The whole video is pretty interesting, but it's a long one. So go check that out. before coming back. You're going to actually see it popping up right above me right about now. And so pause me here and go watch that link. Okay, so welcome back. Hopefully you were just as fascinated about sponges as I was. So in summary, Phylum Porifera is our first divergence from the original choanoflagellate protists. They lack a lot of things. They don't have tissues, they don't have a true digestive system, they don't have a lot of things. But they're still considered an animal. Despite that, again, they're still creating a water current, they're still able to get food, so they still have all the characteristics of animals, but just lacking some of the things that we've talked about, such as germ layers, such as symmetry. So as we explore animals more, you can kind of keep referring back to sponges, be like, well, sponges lack this, but now we see the development of this kind of structure.